abundant coral and sponge communities. We've also seen some really interesting deep sea fish, cutthroat eel. We captured a cusk eel that looks unusual. It might be something new. We've collected a couple of corals as well already. So we were going very slow at the beginning, so right now we're trying to just pick up the pace a little bit and head uphill, which is why we're not stopping to zoom on too many things at the moment. So I think we're going to try to make it to maybe at least 1,300 meters before maybe we slow down for some more close-ups.
Amber, which
Go ahead.
Which seat are you in?
I'm in vid 2.
Vid 2, okay.
But I think you have a summed video, so you talk to both of us.
Oh, gotcha. Okay.
Yeah, that's intended. You did the Atalanta? Okay. Yeah, go ahead now, you'll do the vessel cams.
And science, if you see something you want to slow down and stop for, just let me know and I can stop the move.
Will do. We are going to try to go a little faster than we've been, so we probably aren't going to ask you to stop on anything here for a little while.
Okay. I could just track a line then, if I don't know how you feel about that.
Let's just do individual moves, just in case the slope comes up at us or something like that. You can put in like 30-meter moves or something.
Sounds good.
I'm still trying to remember how to fly. After my nap, I guess, removed all of my brain cells.
Happens sometimes.
Yeah. Yeah, I woke up and I was like, I don't even know what I am, much less like where or why.
Would it be possible to collect a Niskin down here?
Can you stop the ship?
Yep.
What would you like to collect?
Bridge nav.
A Niskin bottle for eDNA, since we are moving up.
A Niskin bottle.
Yeah.
Gotcha.
Stop the current move. Thank you.
And do you want a position over the sponge and the Chrysogorgia? Is that what you're interested in?
More just thinking because we are moving up, right? So we will need one of the bottom one.
Okay, sounds good.
If that's possible.
Yeah, can you take over?
Thank you.
So right now we're about ready to trip one of the Niskin bottles, which we are sampling water for eDNA. We can actually collect water, filter it for
this is going to require some iris manipulation.
Yes.
And can you, yeah, awesome, thanks. Because I'm going to try and see something that's going to be quite dark.
Okay, I can bump up for this.
Yeah, so it's going to look overexposed on all the rest, but she's trying to see the
going,
numbers on the
left.
Little,
yeah, there you go.
There's like these balls. So yeah, you'll want to
And I'm going to start with 1.
Iris is at
even though we don't have...
It's at max right now. Okay.
All right.
1.7.
Gabby, did you say you're going to start with 1?
Yeah.
Great, thanks.
Can you give me the jaws?
So Piera, do you want to tell us and some of our listeners about your eDNA project and what your goals are and how you achieve those?
Sure. So at the Palau International Coral Reef Center, we started the eDNA project in 2021, where basically we collect water inshore and also offshore. Offshore, it's basically we collect a sample in the Palau National Marine Sanctuary.
It's good already? Okay, cool.
Basically 80% of the Palau water are protected and they're called Palau National Marine Sanctuary. That's where we collect water, and then we collect water in the domestic fishing zone, which is the 20% open to fishing. We have already a few data points, and the water we've been collecting so far, it's been in the first meter of the water column, basically, so just shallow water, and this is the first time we have the opportunity to collect deeper than that. So it's very exciting to be able now to collect at 1,400 meter, basically.
Data, do you have that all recorded now?
Because we never did that before.
Yes, got it, thank you.
Sweet.
And we have been targeting fish and vertebrates in general, but once we have the samples, we can basically change the primer and be able to target any other taxa or organisms we want to. So yes, that's very exciting that we're able to do that.
You're
now.
fine, I can line it up now.
Nautilus
We're very
being
spicy.
here.
So when we collect the eDNA, like what exactly are we looking for when we get those samples?
Okay,
so
I think we're ready to keep moving.
So when we collect water, then we bring it back to the ship and we filter it. And then on the filter, basically, we have all the organic matters that stays in the water that from the animal or all organisms living in the water. And we look for what we're targeting.
Can we
so
resume
now our
that move,
fish
30
and the
meters,
diversity
034?
of fish that we can find in the water of Palau, and see what the PNMS is protecting, basically. But it will be interesting in the future to also target other organisms, not just vertebrates. Even we can see sponges, we can see corals, we can see anything, basically, just by changing the primer. So that's exciting. And also it's interesting to see if the eDNA that we collect down here at these depths can match what we are seeing on cameras from the ROV. So that's super exciting.
Go for Zoom video.
Zooming.
You can push in a little further. Nice.
Got a nice glass sponge here.
Okay, go wide.
I'm gonna push past the vignette again.
What's on your mind?
Just wondering if I should reset the DVL.
Yeah, go for it.
All right.
Have there ever been previous dives on this seamount?
We are the first.
Oh,
no one
wow.
has ever dove on this seamount, and until early this morning, no one had even fully mapped this seamount before. So this is great.
Oh, we're the first ones.
We are the first ones.
I think that's what makes it so exciting, especially being in this room, is that not only is it the first time, you actually get to see something that will surprise you, and you don't know what you're going to come across.
Yes. Yes. The terrain's flatter here than the sonar wants us to believe.
Yeah, it's been really interesting. Farther down, we would get these big blocks like this, and they would be covered in corals, but these are not so much so.
Yeah, it's gotten a little bare.
Yep.
Go for zoom.
All the more reason to, yeah, let's zoom on this guy, and then, let's keep moving up.
Yeah, we're keeping ship moves in, so like if I'm zooming on stuff, it's just to kill time.
Awesome.
Speaking of which, should we keep moving?
Yeah, do
Let's
it.
get base.
Yeah.
Bridge nav.
Okay, go wide. That's a beautiful shot, Max.
Same move again, please. 30 meters, 034.
Yep. Really nice shrimp there, a little crinoid sitting on the coral.
You can't see it with the red frame. Okay. That's good. I'm not on rect. Oh. I see what you mean. Good call. That's getting hot again.
Okay, cool. Well, we'll just keep it hot for now. We've had enough practice at that. Yeah, that's fine. Absolutely. Absolutely switch to that mode. Go for zoom.
Going for zoom.
Yeah, so when I'm not perfectly centered up, it's your discretion to sort of pick a zoom level that's going to get things framed up just to get a look at them or something.
Totally. Would you prefer a closer zoom in this scenario?
What's that?
Would you prefer a closer zoom right now?
Yeah, you can go a little closer. And yeah, that's something that'll will sort of like work out between the two of us, or when you're working with Rye, the three of us. Go wide.
Going wide?
When it's appropriate to push in a little further because we've finally gotten the shot centered up a little better or something like that. That does become your discretion. And as we get a feel for it, we probably won't even need to talk about it. It'll just happen.
Totally. I appreciate the advice.
Mm-hmm. Yeah.
Oh, so our pilots, Mika.
Cool, I like that, Amber.
Would like to know, if you have time, how does one become an ROV pilot?
I was kind of looking.
Gabby, you're right.
Sorry, I just realized I wasn't on SPL. I said I don't know how I got here. Like specifically, I don't know where I am currently. What my name is. No, I mean sorry. I don't know.
Just woke up one morning and here you were.
So I personally did a mechanical engineering degree, and then really focused on getting into like the marine industry, specifically like stuff that had deck operations and oceanographic applications. And so many places have ROVs now, so whereas like that used to not be the case. So I think it's kind of easier to brush up with this industry. But yeah, common misconception is like you don't actually have to go to school specifically for ROV to end up in one of these seats up here. So.
Awesome, thank you, Gabby.
Yeah, I would concur. I think the best way to become an ROV pilot is to get like a 2 or a 4-year technical degree, and then just start looking for work that gets you that experience, that gets you offshore. There are a few schools that do really awesome programs specifically for ROV. Memorial University in Newfoundland, Western Michigan, I think, does one. And those are really helpful, and they're especially like well connected and like for internships and things like that. But yeah, I think getting like a 2 or 4-year technical degree is really like the way to do it.
Oh. Well, thank you so much.
Yeah, so that's probably just the issue that we're...
Science, you still happy with this pace? Keep it up?
Yeah, this is great. Keep it up, please.
Okay. Bridge nav. Same move again, please.
That's interesting. So lower came in on its own.
So we're seeing some more glass sponges here. On the bottom left, that was a Bathypathes.
You have more issues with upper? Okay. I kind of feel like I power cycled them, but I almost think that didn't help. Hey, what's up? Okay, heads up next time would be great. Yeah, I think we're just running 2 fibers total right now. Yeah, they're back. We still have choppy lower. Correct. Yeah, we decided to run with it on the last cruise. At the beginning of the last cruise we decided to run like that. I think we had some doubts about that 3rd fiber. It's all good. No harm, no foul. Okay. I haven't been able to get it to work all year. Yeah. Yeah, I'm not, it's the slopes are the same. It's not going to be any good. Yeah, let's leave it for now. Thanks, Dan. Oh, yeah. Hey, Dan.
Dan.
We lost him.
Oh, no.
Go for zoom.
Got a really nice Rutigorgia here.
You can push in a little further.
Pushing in.
Let's see if we've got a little shrimp living in this one. Doesn't look like it.
Okay, go wide.
Going wide.
Nope, just by itself.
What's the name?
Hmm?
What's the name of the...
Iridogorgia.
Iridogorgia.
Yep.
Do you have a common name for it?
I cannot think of a common name off the top of my head. But it's a gorgonian or soft coral.
Oh, okay.
And these ones are characterized by just having this spiral stalk that goes up, and then all the little side arms come off at the top.
Oh, got it.
And oftentimes there's a little shrimp that'll live in the branches on the top.
You 30 meters, bearing 030, please.
You know, I've noticed from what we've looked at so far, a lot of the corals or sponges, they're elongated. Why is that? They're like spread out, far away
so
from the rocks.
they're trying to feed on the plankton and on the detritus, the flotsam that's in the water, and so they try to orient themselves up off the bottom where the flow of the current is a little higher than it would be right on the rocks, right on the bottom itself. So frequently, you see these corals and sponges. They're tall. And they're branching so that they can intercept as many little bits and pieces of plankton as they can as it comes by. And they're trying to trap that in their little arms. So sometimes we see a lot of them on the top, on little high spots.
So write down that the motor's running really hot.
If we
so we think about it.
Come across a boulder that's really quite a bit higher than everything else around it, it'll just be covered in sponges and corals.
Oh, okay.
And so that's part of the reason why I kind of want to make sure we get to the top of this mountain, because I suspect at the very top it's going to be really amazing.
Is there anything specific that we're looking for, or we're just moving
Anything new, anything unusual. Some of these animals we have seen before in other places, but some we have not. Some we may not recognize as being new until afterwards when we're going through the video again.
Oh, wow.
But yeah.
Okay, here's a challenging one. Go for zoom.
This is a great sponge right here.
Going for zoom on the great sponge.
These are tough to get your iris right on.
Yeah.
Wow, that's so interesting.
Stop there.
What is...
Nice
That's
job.
great. So these sponges are drawing in water from all around the outside of the periphery, and then that water is then forced up through those center openings.
Okay,
we
go
can
wide.
see 3 of them right in the middle of the
Going wide.
sponge, and that's where the water comes out. And the sponges actually filter out particles that are way smaller than what the corals can filter out. And we now understand that these sponges can actually absorb dissolved material. So sugars or other compounds in the water, they actually can feed on that stuff. It's incredible.
Wow. It's amazing.
Okay.
Do you have a guess for what kind of sponge
Can
we
we
were
just-
looking at?
What is that? I am not sure what kind of sponge that is. We are trying to look that up right now. It is probably in the genus Sacocalix, but it's definitely a euplectellid, but
Go for zoom.
a glass sponge, but probably Sacocalix, but we're not certain.
Science, do you want to keep the lasers on?
We can turn them off for now if you want, but it is useful to have them on if they're not bothering you.
Oh, they don't bother me at all.
Okay, great.
Okay, go on.
Going wide.
What are the lasers used for?
The lasers are basically a ruler. When you're looking at a single video screen, it's very hard to have any size perception. So when you zoom wide, everything looks really small. When you zoom in, things can look really big. And so the two green lasers are 10 centimeters apart, about 4 inches, and it's just a ruler. And anywhere we look, we've got a ruler placed over the objects that we're, so we can tell how big things are.
Can you look back at a previous dive and see what we were running the motor at?
Bridge nav.
Yeah, that's what I thought.
Ship move 30 meters,
Yeah,
bearing 030,
never
please.
see it at 70. There was one dive, and that was the one where we wrapped the motor comp. The super shallow one in like 20 meters of water, or 200 or 100 meters of water, 26 degrees or something. The comp's not going up though. Yeah. Okay. Yeah. Okay. Yeah, I think when we sit and sample is when we're gonna see it go up the most.
And here I'm hogging this, but
Go for zoom.
Going for zoom.
this is another Bathypathes. It's a black coral. We've been seeing quite a few of these, and we sampled one of these, so we'll be able to confirm the identity of this taxa.
Okay, so is the Bathypathes the thing that's in the front box that's floaty?
Yes, that is correct.
Hmm.
Uh-oh. What are you telling me, Gabby?
Oh, it's just, I've had more than my fair share of corals with Bathypathes in the front box.
Yeah.
It's my nemesis. Yeah.
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 They float, I presume, so that they're always sticking up off the seafloor and filter feeding, but it's very inconvenient when you want to sample one.
Yeah. They didn't really think about us.
No.
In their
really rude of
design.
them.
Yeah, so rude.
Go for zoom.
Zoom it.
Yep.
So this looks like a Metallogorgia. It's related to that Erigigorgia, but instead of being a little spiral, it just has a series of plumes on top, and then every single one of them has a brittle star that lives in the middle. And it's a little symbiosis with a coral. And exactly the nature of that symbiosis is still a little up for debate, but
Pull that.
brittle star probably helps the coral and vice versa.
How small are the brittle stars?
The brittle stars probably have a disc diameter. Well, you know, even when we see really small Metallogorgias, they already have a little brittle star in their arms. So this starts very, very early, and presumably they grow together. So you'll see the one on the top right, that has a brittle star in it as well. Every time we see one of these.
That's amazing.
And then on the bottom here, this is a lyrate coral, and I'm not sure which one it is.
You said lyrate?
Yeah, so it's got this, like, looks like a menorah or a candelabra or something with all of these little arms sticking up.
Awesome.
Can you look?
Could we turn the lasers off for a second? Get a photo of this?
Yep.
Okay. Wow.
Awesome. Thanks. Okay, go wide.
Going wide?
Mm-hmm.
Do we want lasers back on, Science?
Yeah.
Yeah.
Sure, that'd be great. Okay. Some sort of calypto. That
let some of that
that lyrate one was probably some sort of calyptophora.
The next move.
But this is a fish guy talking about corals. So if anybody online wants to correct me, please go for it.
It's not a lot, but I can stop and look at some stuff. Oh, this is a nice one right here. Go for zoom.
Zooming.
Okay, go wide.
Going wide.
I was just imagining her turning on the AC in the HPU. Oh, there's a Victor Gorgia under there. We haven't seen one yet. On this watch anyways.
What was it, Gabby?
Victor Gorgia?
Oh, yeah.
Yeah.
Okay, you can keep moving.
Okay.
That's okay. Yeah.
Bridge nav, can we do 30 meters bearing 022?
Science, we're at 1364. Do you want to keep up this pace?
Yeah, we're going to keep it up till we hit about 1,300 meters or so.
Sounds good. Things are starting to get interesting again.
Okay, and we've got some feedback from Asako, who's online, and that was Calyptrophora or Paracalyptrophora that we saw, and then some of the unbranched ones, the whips have been bamboos. Thank you, Asako.
It's so great to have so many awesome scientists participating from shore as well and contributing their expertise while only a few of us maybe can be at sea in the control room. It's a pretty cool opportunity to be able to engage with people from all over the world and bring in that expertise.
Oh, that's a great sponge right there.
You like the sponge? I really like that Iridogorgia.
Yeah.
Okay.
Let's zoom on that guy.
Video. Let's go for zoom on this sponge.
The Iridogorgia is really cool. We've seen a bunch of them. But
oh yeah, there's trillions of them. They just happen to be my fave.
Yeah, they're incredible.
They're very charismatic.
They are.
Know, as deep sea corals go.
Yep.
What can you tell us about this sponge, Jeff?
Nothing just yet.
Sweet.
Hey, Science, for that first Niskin, what sample number was that? Or what do you want to call that one?
Noel?
It was 006, NA167-006.
Thank you. I'm just putting a target in HiPAC.
No problem,
I
thank you.
noted when we dropped it, or collected it. Thanks.
Could we also turn their lasers off for a photo of this beautiful sponge?
Yeah. Cool.
Can you scroll?
This is starting to make sense.
It is a gorgeous sponge, but we are not sure which one it is. Yeah, some of these other things that are to the right, those are probably the bases of other sponges that have died. Potentially corals as well, but I think the one to the right looks like the base of a sponge. And it's really interesting to think about, you know, we see these, and oftentimes we don't see very many super small sponges. And so you have to wonder, like, how often does a new baby sponge start growing and recruit into the population? And how long do these things actually live? And there is a fish in the background above the coral.
Are we switching to fish mode?
Oh, hell yeah.
Okay.
Fish mode.
Go wide.
Going wide.
Oh, it's really far away. Or it's really small.
I don't see it anymore. Where'd it go?
It's a smart fish.
Yeah. Yeah, Jeff, I feel like we're fishless now.
Oh, well.
Okay.
Let's
there
keep
will
going.
be other fish.
Yes.
We're still
there are
we're
many
keeping
fish
the
in the
moves coming
sea.
here.
Yes.
Do you want me to wait for Atalanta to catch up?
Oh wow, yeah, we're for whatever reason just dragging more now, huh?
Do you feel any current right now?
There has been some. I don't have to work very hard for it, but there's a bit.
It kind of looks like still swinging.
Or it's just gonna sit there. I don't know.
All right.
Is it moving?
A little bit.
Okay.
It's still poking forward.
I can get moving for it a little bit more too. Can you give me a reset?
Yep. All right. Seems to be stable where it is.
Cool.
Bridge nav. We'll do a 30-meter move at bearing 022.
Go for zoom.
Going for zoom, sorry about that.
Nice job. Nailed it. And then as I move, the lights change position relative to the sponge.
Yeah, sorry about that. I keep inverting the white balance iris.
It's totally cool.
In the wrong direction, but
that's a really nice looking sponge.
that's a lovely
Okay,
sponge.
go wide.
Going wide. realize it was already a few minutes ago, but did we have an ID on that little crustacean in the sponge earlier?
I do think the current's changing a little bit. The tether's lying in a different spot, but it's not very strong.
Okay.
Copy that. I appreciate it. Okay, how's that? Okay.
How long is this dive still going to be? Well, the dive plan right now is about 14 hours. How long have we've been diving?
We dove about 2 in the afternoon, and I think we go until 4 in the morning, local time.
So we have a few hours left of this dive, but a total of 14 hours.
Just do it. Do it.
Yeah, we have roughly 6 hours, I think, on bottom. About 6 hours more bottom time.
Okay. Are we making progress at any sort of reasonable rate?
Yeah, we're doing great.
Okay.
I mean, on this watch,
It's
we are.
another amazing.
Wait, did you guys already get to a checkpoint?
Yeah, we're pretty much at waypoint 3.
Oh man. We're a little
What I'm
slow
hearing
pokes.
is we have like not stopped to smell enough roses.
I think we're going at a great pace.
Yeah.
Jeff wants
Go for
the
zoom.
fish.
Jeff's a peak bagger, I think.
Yeah.
Peak bagger.
Oh, I love this
cool.
sponge. Oh my gosh.
Wow.
That
What
is
a
really, really cool.
fantastic creature.
Do you want to get
This is
even
another
closer to it?
Saccocalyx-like sponge.
Feels good
I'm
there.
not,
Because she's
it's a little
moving,
different
it's a little
looking,
harder.
but very similar to that.
Okay, go wide.
Go wide.
And then we can turn lasers back on. Hey, science?
Yes, that's fine.
These sponges are just really charismatic.
So cool.
Oh yeah, we should give some more credit to black corals just because they're not like big and shiny. Go for zoom.
Zooming.
Bridge nav. Same move, please.
I am
30
not
meters.
sure what that
0,
is.
2,
Okay,
3, 0, 2,
Sacco,
2.
if you're
Go
online,
up.
what are we looking at right now?
Yeah, all I got is black coral.
Yeah, these, no.
Does anyone know much about the geology of this region?
It's a little dark now.
I know only sort of the broadest concepts here. Basically, we're at a plate boundary. So to the east of the islands is the Palau Trench, and extending north of that is the Yap Trench, and then that goes into the Mariana Trench. So we're in a place where there's oceanic plates that are diving down beneath the plate that we're on right now. And as a result of that, there was a lot of volcanism. This started millions of years ago, and that's what created the Kyushu Palau Ridge that the islands of Palau sit on. And so those were well above sea level at one point in the distant past, and a lot of coral grew on top of them, and then sea level has changed, and that's what created the rock islands, is it's all uplifted coral reef. So in this place, I presume it's another volcanic peak that formed from the same processes that the main islands did, because we're due north on that larger ridge feature.
Is that motor comp still climbing?
No.
Okay.
Yeah, it's been at 8 for the last couple hours.
Okay, cool. Go for Zoom video?
Go for Zoom.
You push in a little further. Nice.
Oh, that's great. You can see all the individual polyps.
You can play with your focus a little bit.
Thank you. Appreciate it. Yeah, it's a little...
Like sometimes you have the back polyps in focus, sometimes it's the front ones.
I see, I see.
That looks awesome. Nice shot. Okay, go wide.
Nice.
Go wide. Appreciate the patience.
Oh yeah, you're doing awesome.
Yeah, good job, Max.
Do you feel like I'm gonna smack your head whenever I do that? Because it feels like I'm gonna smack my head.
I definitely bit down a little.
Yeah. Sorry about that.
It's okay.
I mean, it's not actually going to impact your head, but it does feel that way. Can you come up a little bit?
Oh yeah, I am actively coming up.
Okay, there's a fish right there.
Okay.
Just click.
Go for zoom.
Yeah.
Zooming.
I do not see this fish.
I think it's
It's
under the
right
overhang.
underneath that little ledge.
Okay. Oh, sneaky.
Uh-huh.
Oh,
Look at that.
there.
Yeah.
Okay, this is some kind of cusk eel. Gotta zoom in here.
Okay, I'll get a little closer.
I'll zoom a
Go
little
for
bit more.
zoom. Gonna have to play some games here with.
Oh, here I can unspecify that.
Yeah, that's not so good. I need to like look up and go down and yeah, I think I need to be looking up here. There we go.
We want to keep zooming in on that, or?
Yeah, go for a little more zoom.
Ship's not moving now, by the way.
Cool, thank you. Jeff, are you getting the imagery you want?
Yeah. Can we zoom at all anymore? I know we may be maxed.
Yeah, definitely, but yeah.
Okay.
Go wide for a sec.
Fully wide?
That's full wide.
Do you want me to go full wide?
Yeah, go full wide.
Oh, that's great.
Go for zoom.
Zooming.
Just go all the way in.
Oh, that's fantastic. Okay.
You got your image?
Yeah, this could be Diplocanthopoma.
Nice focus.
But this
really
is really, really good. You can see the pelvic fins are just a couple of little isolated rays right underneath the guy's chin, right below the pectoral fins.
I haven't sailed with a fish person before.
I am not exactly sure who this guy is, though.
Should we get the net?
Underneath that ledge, I think that would be impossible.
Fair enough.
Gabby, if you think we can get this fish, I mean, I'm not gonna say it wouldn't be a bad idea, but I think it's pretty inaccessible.
It doesn't jive with the strategy you described.
Indeed. Yes, I would agree.
Needs the fish to be a little bit more out in the open.
Yeah, agreed. This video is great, though. Let's.
Okay.
Awesome. Thank you.
Yeah. Nice spot. I don't know how you saw that.
That was impressive.
That was Piera. Piera's got an eagle eye.
Oh yeah, nice one. Okay, are you happy with the images in the video that you have here?
Yes, this is great. Thank you.
Go wide.
Okay, going wide.
Oh. That guy right there.
Okay. Let's see if
that's some sort of shrimp. It could be Nematocarcinus.
Zoom video?
Go for zoom.
I have to get up. Yeah, go wide. I have to get up. I just got a little lazy there. I know better.
I'm just gonna do a round of gauges while we're looking at things.
Okay, sounds good.
Want me to start moving again?
That is a question for Jeff. Science, are you ready to start moving again?
If we can get as one zoom on that shrimp, that would be great, and then yes.
Yeah, I'm just fighting with the tilt functions here. Okay. Okay. Go for zoom video.
Zooming.
Yep, okay. That we've seen a couple of these already. This is one of those really cool shrimps in the genus Nematocarcinus. They have legs that are 1.5 to 2 times their body length. Wow. They can spread out all of their sensory appendages.
You can push in a little further. Nice.
Better search for food and keep on top of the sediments and things like that, but this guy is just crawling around on the rocks.
Okay, go wide.
Pull back.
There's
Science,
a really amazing coral immediately to the right, too, that we might want to take a quick look at.
Okay. Oh, wow. I don't know how I didn't see that. Is that a coral?
I'm not sure. My first instinct was it was a crinoid.
No, I'm going, I have to guess coral.
Yeah.
Go for zoom?
I think
we'll see.
Yep.
Zooming.
Oh, that's beautiful.
Can we turn the lasers off?
Yeah. You got it?
I got it.
There's an anemone on it too. Why can't I say that word?
An anemone.
Anemone.
Really look like pine needles.
This is really beautiful.
That's great.
Okay.
Thanks.
Okay, go wide.
Going wide.
Bridge nav. Do a 30-meter move bearing 021.
Isako said that is a crinoid.
Sweet.
Okay.
Oh, wow.
With branched arms.
Yeah.
That's incredible.
Does that mean it would move?
I think that one is not a moving one.
Okay.
I think the stalked crinoids don't go anywhere, but I could be wrong about that. There's probably one stalked crinoid, like one really obscure stalked crinoid that like jumps and tap dances or something.
Tap dances.
Well, I always thought all anemones were sessile, and then one jumped, and it turns out that there's a jumping anemone.
Really?
Yeah, absolutely, 100% jumping anemone.
I didn't think I'd need this jacket until going back to Boston, but here we are.
Go for zoom. I'm looking at the unbranched coral right in front of us.
The pipe cleaner looking thing?
Yeah. Keep pushing it.
Yep, okay, that's one of the black corals. Yep.
Nice. I guess it is branched, it's just branched at the bottom. Can you push in a little further?
Absolutely.
Okay, go wide.
Go wide.
Mind if I reset the DVL?
Go for it.
Gauges are good.
Sweet.
Yeah, motor has not changed.
Yeah,
so
okay.
So we're just running hot. And that went down again. Yeah.
Yeah, I guess I'll just let that go and see if it comes back. Yeah, there we go. Go for zoom.
Zooming.
Great, right there. Nice. Max, you definitely stuck the landing on that one.
Thank you.
Go wide.
What is that moving around in the center there?
Go wide.
Jeff, is that a coral?
Go wide.
Yep, that's another Metallogorgia, the little brittle star in the middle.
Ah.
Okay, I have a great question from one of our viewers. This is about sampling. So they asked, have you ever thought that by picking up specimens, you might be interfering with evolution? Descendants, for example, like our family tree. Have you ever? That's a great question, by the way.
Yeah, we do not sample lightly. Oh, there's a fish. Okay, I will come back to that answer momentarily. But we have a cutthroat eel here. If we can get a zoom and we can take a look at the fins, we might figure out which about one of four species it is here.
Zooming.
Oh, wow. Stop
Okay.
there.
Yep, it's in the genus Synaphobranchus. And if I can just get him to run into a rock and stop for a second, he might flare out his fins, just like that. There he is. Okay, that is Synaphobranchus oligolepis.
Oh, you got it? You got your
did.
ID, hey?
I got my ID. I just needed him to flare up his dorsal fin there. And it starts really far forward, kind of towards those pectoral fins, so it's probably Synaphobranchus oligolepis.
Okay.
Very cool. Thank you.
Uh-huh.
Going wide.
Awesome. Do you know the common name for that?
They're called cutthroat eels, but the
eels.
individual species don't have common names.
Oh, okay.
Yeah,
Okay.
so that's the name applied to the whole group.
Okay.
Yep.
Gotcha.
Yeah, what's up?
So back to the question
Absolutely, go
of
for
the caller.
Can you leave the upper one?
We do not sample
Beautiful.
lightly,
Thank
and
you.
we do not want to sample a lot
Appreciate the heads up.
because of those reasons. We don't want to interfere with the population at large. Yes, we are sacrificing a few individuals so that we can learn a whole lot more and hopefully through that knowledge be able to achieve grander conservation goals. But we are typically only taking a couple of animals in an entire dive, and it would only be a couple of animals probably for over a decade. So it should not affect the population at large.
It's much better.
But it is sacrificing an animal. If, for instance, we're sampling a fish.
What
For the
magic
corals,
did you
we will
just
take
do?
a branch of the coral so that we don't destroy the whole colony. So we only take a small piece of it.
Beauty. Love it.
I hope that addresses the question, and if not, hopefully they can type some more.
My life just got like very measurably better.
What happened, Gabby?
The lower camera isn't choppy anymore.
Oh.
Very distracting, and now it's beautiful.
I thought Jeff just gave a really good answer.
That too.
Yes, also
I did
that.
like the fish whispering where
Ridge
you
nav.
told him to run into a rock and then he did.
30 meters, bearing 024, please.
So when we get to 5, are we just sort of in explore around mode, or?
Yeah, I mean, it's kind of downhill from there.
Oh, bummer.
But lightly, like it's not steep.
Oh, okay.
Yeah, we can slow down a little bit now, but
Okay.
I just wanted to make sure we were going to get to 1,000 meters with an hour or two to spare at the end, and I think we're definitely going to do that now. So,
Okay.
So you can see. at the top.
Brian in particular wanted to make sure there was time to explore the top of the seamount. So,
Is waypoint 5 the actual top?
Yeah,
Okay.
yeah,
that's the high point.
and it's pretty shallow slope up there. Rennie was
Mhm.
saying it's less than 10 degrees, so hopefully it wouldn't really be fully downhill if we were exploring around up there.
Yeah, that ridge to the east, northeast looks, it's pretty mild slope.
Mhm.
Have we collected any rocks yet on this dive?
No, I don't. Let me double check, but I don't think so.
Yeah, I don't think we have. Lower down, we were worried that the pieces that we could actually pick up were just going to be talus that had fallen down slope. So if we see an outcrop where we think we can take a piece, it's not like totally cemented in with the manganese crust, we should probably do that. And Gabby, I don't know what is something we can grab or not.
Okay. I'll keep an eye out. I mean, this stuff's all really cemented, but
Yeah.
I'm often surprised by what you can break.
Okay.
So, I don't know. Yeah, we'll keep our eyes peeled. Or something that looks like it's broken in situ, like where it's very obvious where the parent rock is.
Yeah, right.
There's some sort of eel thing in the camera of the umbilical.
There's some sort of what thing?
Oh, eel thing in the umbilical camera.
Oh.
It's kind of cool. Zipping around.
I missed it.
You'll see him again. He's just circling.
Go for zoom.
Oh, that's a cool crinoid.
Zooming.
I've been doing all these cruises with coral people, and they're not that into crinoids.
Crinoids are my chaunacops, maybe. I really like them.
You really like
I
really
like
how they
them.
move. I haven't looked at a lot of crinoids recently. I do like them. Also, not that many fish. And Megan likes corals and sponges.
Yeah.
Spent a lot of time with Megan.
She can ID them all almost.
It's unreal.
It is unreal.
And she can ID them from when they're the size of a pixel on the screen. There's like a pink smudge, and she's got it to species ID. Yeah.
There was one dive that she did up off Alaska, annotated, and I think it was something like, in one dive, it was like 20,000 annotations.
Oh my gosh.
Yeah, she looks at a lot of corals and sponges.
No wonder she's so good at it. I love these things when I'm not trying to collect them. They're really beautiful.
They are.
Bathopathes. Go for zoom.
Zooming.
They're so pretty.
Symmetry is amazing.
Yeah. Okay, go wide.
Going wide.
We are coming up to a cliff.
Are we?
I don't know. I was just looking at the sonar and looking at this rock ahead of us.
Oh, the sonar's kind of been looking like that. This is like very, 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 very reflective terrain.
Okay.
So
it's not a cliff. It's just a slope.
No, it's more of this, I suspect.
Yep. Let's go take a look at that bamboo or that lyrite coral over there on the left.
Okay. Go for zoom video.
Zooming.
Yep, we've seen a couple of these so far. Okay.
Bridge nav.
You can push in further.
Can do.
See if you can go all the way in and see if you can get some polyps.
Move, bearing 022.
Get a focus on them.
That is all the way in.
Often the polyps being in focus is what gives them the ID. Nice job.
Asako says this may be the Bathopathes pseudo-alternata. But she's typing again, so maybe she'll update us.
Okay, go wide.
Going wide.
Are we moving too fast now? Like, are we going to get there too soon?
I think it depends how much time we want at the top. I mean, it will get steeper after waypoint 4.
Okay.
So that could be pretty slow.
Anything in here look like we could sample it rock-wise? Just thinking of something to slow us down.
Oh, yeah, okay. Well, there's... No. No. No.
It's all pretty solid.
Bedrock?
I mean, there's these little fins here. Oh yeah, there's not really, no, they're not what I thought they were.
Is anything like that up there?
Go take a look. I wish we had one of those rock scientists.
One of them geologists?
Yeah. Tell us which rocks are the breaky kinds of rocks.
Uh-huh.
Maybe this little corner here, actually.
That right there?
Yeah.
Yeah.
We'll give that a go.
Sure.
Oh, that is spicy, sorry.
Oh, hey, look at that.
Nice.
Hey, that was more successful than I anticipated.
That was great. Okay. Rock sample number 1. Hold on. If you could keep that steady, I'm going to take a photo with the
Here, I'll bring it
camera.
up. I'll
Great.
bring it up into the light and and rotate it and stuff
Awesome.
like that. Okay.
So is this sample 7?
007, yes.
Thank you.
Noelle, is it okay if we put this in the starboard box?
Yes, that'd be great.
Okay. And where do you want it? In one of the small ones, I think, preferably.
Yep, one of the small ones. The first one would be fine. A is fine.
The most forward, is that the
Most forward box is fine. Or
Okay.
whatever is easiest for you. There is one sample in the third
Okay.
bio box.
Oh, the third one's the best one.
What is the sample in the?
Yeah.
In that?
That
okay.
is a
Can you set me up for that?
Yep.
Starboard box out. Give me the right
pink
camera.
Paragorgia is in the third bio box. The rest
Okay.
of them are open.
That's really, really porous.
And then, can you give me samples?
sample?
Yeah, awesome, thanks.
Nice.
Thank you.
Great. Ryan, I'm interested to have you try the topside controller in this position.
Oh.
I don't think I'm a huge fan, but I
Is
think
that
it's,
what Dan runs at?
I guess so. Yeah, you can close up and
Yeah. You want dive special back?
I can do it. Got it. I don't know whether it's Dan who did it or Jake. But yeah, it's strange. I haven't spent a lot of time with it like this.
Now that we've got our rock sample, so what are we going to do with it once it's on board? Do we dissect it or?
So the rock will ultimately be used by people probably not on board ship to date the age of the seamount. So they can use the chemistry of that rock to estimate its age. It kind of looked like carbonate to me, so I don't know. I'm a little mysterious. I thought perhaps it would be much more like volcanic rock in and maybe it is, I'm not a geologist, but it seemed very light inside. But it should provide some clues about the origins of this seamount that we're diving on.
Gotcha.
Yeah.
Should we move on?
Yeah.
Yep.
Yeah, shall we?
Yes, please.
Bridge nav. Please do 20 meters bearing 022.
Motor just got up to 76.
Oh, okay. Yeah,
Yeah.
just from sitting still. Let's see if it goes down.
Yeah.
Yeah, do you want to drop a note in the chat about that?
Yeah, sure.
I know both watches have seen it and not been super concerned, but
Yeah. Sorry, let me
yeah,
just
it's
get
coming
on
right
top
down.
of this now.
Mind if I reset the DVL?
Go for it.
Okay. Yeah. Thank you. Okay, I'm back. Yeah, sure. One viewer says that these are not volcanic rocks. Everything that we're looking at right now is carbonate. That's interesting. Well, thank you.
Bridge nav. Same move, please. 20 meters, bearing 022.
I just looked at the port valve pack and saw the comm status at red and had a moment of like, what?
I did that earlier.
For the thing that I said to do.
Poor port valve pack.
What's that?
Poor port valve pack.
I know.
Has to sit this one out.
Yeah, buddy.
This terrain really has changed, and it is really barren.
It's really what?
Barren.
Yeah.
There was a lot more lower down, a lot more life.
We've come such a long way.
What is?
I'm going to do one more round of valves on the hour.
Okay, sounds good.
And then that way
oh, that's kind of you.
you can, it's actually purely selfish, so that I have your undivided attention to reteach me how to fly.
I've never been very good at doing gauges and other things.
Yeah.
It's not my forte. Especially if I'm not doing it 4 times a watch. Like, I have to hunt for everything.
Yeah.
Rye, if you look at that sonar, I forget how much sonar stuff we've talked about. How do you know that's not a cliff?
Because it's not all solid purple. Like you have stuff going off into the back versus like a thin line of, yeah.
Good. Good one. Nice.
Cool. No garbage.
Yeah, like in this base.
Oh wow, look at this. That's cool. This is cliff-like.
Usually a spot like this would have a ton of life.
Yeah. If we just pan to the right really quickly, we're not missing something on the corner just right next to us or anything, are we?
Coral. Up ahead there is a single, maybe like Chrysogorgia or something like that.
All
Oh
right.
no, it's a dead sponge. Even worse.
Yep, you're right. Okay. Yeah, you'd figure on this left edge that there would be a lot of corals and sponges.
Is that what that is? A dead sponge? Or no, it's a Bathypathes.
It's Bathypathes.
It's terrible. That's like the opposite of Megan.
Megan's probably rolling in her bunk.
Yeah. Why did that take you 3 chances? And you went from coral to dead sponge.
We're giving her nightmares.
Bridge nav.
Go wide. Oh, I guess I didn't have you zoom. I just went really close. I can pull back.
20 meters, bearing 023.
Thanks, video. You can push back in past the vignetting. Good.
The rock is very close.
Yeah, I've got the porch on it.
Cool.
I did my own zoom. That's what that is.
I couldn't tell if you were like porch under and
I was porch under a little bit.
Manual zoom.
Boop the WCA. But
So Jim Reimer is typing in the chat here a little bit. Was just curious about whether we had been seeing any garbage or nets, which we have not been seeing. And he was saying that he had done some work north of here, but still on this Kyushu Palau Ridge. And some of the seamounts to the north were scoured with all kinds of nets and other garbage. So really kind of an interesting contrast that here,
fun.
Yeah, but really an interesting contrast here that we don't see that. So
are they in the National Monument as well? Or the Marine Sanctuary? Sorry.
I will ask.
Because I wonder if, while we stay inside the Marine Sanctuary, whether we're sort of free of that.
Exactly.
Sort of partially out of self-interest.
He did say that those dives were shallower, so it may be we need to get to the top before we can make real comparisons. But
okay. Sorry, video, did I have you zoom in there?
You did.
Can you go wide?
Chris Kelly has also joined us, and he is confirming that this does not look volcanic, but this looks like manganese-crusted carbonate.
That's kind of what that rock we sampled looked like.
Yeah, it definitely, when we broke it off and it was white inside, yeah. And that it broke so easily, yeah.
Go for zoom.
Zooming.
Jeff, was there something that you wanted to circle there?
Yeah, there was a coral there.
Go wide.
We can zoom on this guy right here. That's fine. That's fine.
Yeah, we'll do this coral and then we'll do the other coral.
Steve Oskovitz is online now, and I know he wants some close-ups.
Okay, awesome. We can do that for Steve. You can keep pushing in.
All right. Just wait for the go ahead.
Okay, go away.
Chris is saying that this is Primnoid, possibly Calyptrophora wyvili. Oh, great urchin right there too that we just went past.
The white
yeah,
urchin,
let's check out that guy.
Okay, zoom.
And I am being corrected. The pink coral we just looked at is hemicorallium, which makes more sense because Megan pointed that out earlier in the dive.
You can push in a little more.
Oh, this is a great urchin here.
Oh, nice focus there. That's beautiful. Push in a
Yeah,
little more.
that is great.
Back soon.
A spitodiadema.
Can we turn the lasers off for
All
a second?
pushed in.
Yeah.
I got it.
Okay, go wide.
Go wide.
And then, Jeff, you said you saw a coral.
There's another coral up to the right.
This yellow one?
No, there was another one.
Okay.
I have to zoom out a little bit more. Right there.
Okay, I see it. Go for zoom video.
Zoom in.
Black coral. You can push in further.
Push in.
You can go all the way and try and get a
are
focus on the polyps.
all the way in.
That's all the way. It's not very stable here. Possibly because I thought I pressed stick lock, but did not. Okay, go wide.
Go wide.
Anything else you wanted to look at around here?
Let's keep going.
Okay. I think we're going to do a
nav.
quick swap up front here.
20 meters at 023, please.
I really like this rock.
Yeah.
That's
like
cool.
A hole down the side.
Here, we'll perch on it. Just a small perch. And can you correct the speed? And we'll go 2.2 for now.
Say that again?
The speed of the moves, switch it to 0.2.
Okay. Bridge nav. Could you modify the speed of this move to 0.2 knots, please? Thank you.
What we got float by there?
You're just going. Yeah, get yourself centered up in the Atalanticam. To the extent that makes sense. But I wouldn't do it by going too far ahead, more by moving laterally. You're sort of stretched out already.
Jeff, why are you Daniel in the Scientists Ashore chat?
Because Daniel left his login open and I'm using it. So all bad IDs, those are Daniel's.
Yeah, so we're moving slow, just feel it out. The slope is still coming up ahead of you. This is just very irregular terrain. Yeah, absolutely. Do you remember where to look on nav screen? Okay.
Oh, there's a fish on the right.
They're cutthroat eel.
And chasing fish is like the #1 way to get yourself sort of backwards, turned around, and off in the rhubarb.
Oh, there's another little fish next to it. But there's a little tiny one right there, but let's stick to that guy.
Go presume.
Okay, another Snappabranchus or cutthroat eel. If we can just keep trying to zoom on this cutthroat eel, that'd be great.
Okay.
The other guy was too little, he's probably gone.
Keep zooming, right? Little shrimp swimming by.
Oh, little shrimp there. That's probably a Canthophyra, a little red shrimp.
Absolutely.
Take a look at your altitude as well.
Go wide. Squat lobster there.
You're in auto head. Yep, it sure does.
It's definitely Snaphobranchus, but I cannot tell which one. We may have lost him underneath the rocks there.
Go at it.
There he is. I think we're gonna take a look at this coral to our right. Immediately to your right. Nope, keep going a little bit more to the right and look a little bit down. Yeah, let's see if we can look at that chirostylid and the coral there.
Zooming.
It's a black coral. I'm hoping the guys online know which one this is.
Thank you.
Yeah, thanks. So we can zoom in on that bright orange one just to the left if you want. Yeah, looks like a Hormathiid anemone, Venus flytrap anemone. And that black coral looks like it was a Bathopathes.
Zooming. Pull back a little to zoom.
Those guys cool.
That's crazy looking.
Yeah. They call them Venus flytrap anemones, but it's a Hormathiid. Okay, let's keep going.
Ready to move? Alrighty. Bridge nav. Could we please do 20 meters bearing 020 at 0.2 knots?
That cutthroat eel from earlier.
Oh, yeah. Yeah. Yep, we already got a good look at that guy. I'm noticing a lot more little shrimp in the water right now. About 3 or 4 have just passed by. We're getting shallower, and I wonder if we're getting into that layer where the abundance of water column animals is going to be hitting the sides of the seamount now. We've got a couple little sponges and some more crinoids on these walls.
So in the earlier part of this dive where we were seeing more biology, was that, I kind of missed it, but was that a basalt rock? Or?
Yeah, I think it was. And yeah, it's hard to separate whether depth or current or substrate type is driving the differences we're seeing because they're all changing at the same time. Yeah, I was wondering the same thing. What's that? No. I think we got some sort of echinoderm here. Yep, looks like we got a cushion star.